so much for coming. Just out of curiosity, can you hear me okay in the back, even if I'm not right next to the mic? Okay. So I'm so excited to be here. This is a really unique opportunity for me. And I'm really excited about the opportunity to talk to people who think about some of these concepts and topics in a very different context than me. So I want to just jump right into the symbiosis or the interaction I'm going to be talking about today, because there's just some like bonkers cool biology going on here. So what you're looking at on the slide right now is a plant root really close up along here. And then these pink structures are what are called root nodules. And these root nodules are built by cooperation and back and forth dialogue between a plant and a bacterial symbiont, so a microbe. The best way that I can describe these things to you is that what you're looking at is a marketplace. So essentially what happens in these nodules is the exchange of resources that I'm going to talk about more in a second between the bacteria that are resident in the nodule and the plant that's hosting the bacteria. Because of this, because of interactions like this one, but not limited to this one, plant microbe symbioses have been developed as model systems for thinking about communication, cooperation, and negotiation in biology in a bunch of different contexts. And a lot of what we learn here, we can extrapolate outside of plants to animal systems, but I think it also is a good model to understand trading and things like that in general. So a bit about this particular mutualism. This mutualism involves the exchange of carbon and nitrogen. So the bacteria that live inside these nodules are nitrogen-fixing bacteria. They are what I'm going to refer to throughout the talk as rhizobia. This ability has actually evolved independently several times. There are several genera of bacteria that can do this. All of them live inside nodules on legumes, which are plants in the bean family. So while this ability has evolved multiple times in bacteria, it only appears to have evolved once in plants, at least in this particular and specialized way. So when I say legumes, for those of you that don't think about plants, a legume is something like a soybean, a pea, a green bean, things like that. There are a couple things that I just like to share to contextualize why I think this symbiosis is really cool. One is that this is why beans are such a good source of protein. So nitrogen is a major component of proteins. One reason that we think that beans are such a good protein source is because of the extra nitrogen that they get from these bacteria. The other thing that's quite cool about this system that has nothing to do with what I'm going to tell you about today is that these nodules are not false color in this image. I'm not showing you a false color picture here. These nodules are actually pink. And the reason that they're pink is the same reason that our blood is red. Legumes make a molecule called leg hemoglobin, which is analogous to animal hemoglobin. And it's performing a similar function. It's an oxygen shuttler. And so it's controlling the oxygen concentrations in these structures. As an aside on that aside, leg hemoglobin in these nodules is why Impossible Burgers taste like meat. So the plant-based meat industry has capitalized on mutualisms like this really recently in a really cool way. Okay, so this mutualism itself is quite cool and well studied, but it's by far not the only thing that works like this. There are thousands, probably millions of other resource exchange symbioses like this one in other plants, in animals, and in humans. And so this is a really excellent model for a couple of reasons, but it's not a unique or aberrant example. So that makes it, I think, a really good case study to ask some cool questions. And the questions that my lab focus on generally fall under this theme. So what happens when communication, cooperation, or negotiation in an interaction like this are either intercepted or disrupted? And what I'm going to tell you about is primarily the role of parasites, which is what my lab has focused on a lot so far. I'm going to talk at the very end about some other sources of interception or disruption that we're starting to work on too. So before I jump into the data, I just wanted to give you some context about who I am and where I'm coming from. So I'm an evolutionary ecologist. I study how organisms change over time. I'm especially interested in how their environment influences how they change over time. My undergrad training was not in science. I have a BA in English. I actually discovered evolutionary biology because of an interest in comparative linguistics. And so I'm really excited to be talking to folks who think outside of biology. And I was really excited to come here because I'm interested in how other fields sort of approach or frame or investigate some of the things that I think about in biological systems. So the other reason I wanted to say that is that if I'm lapsing into jargon or talking about plants or evolutionary concepts that are unfamiliar, please pause, raise your hands, and ask for clarification. And interrupt with questions if you've got them at any point. Okay, so when I think about the establishment of a symbiosis, when I think about the formation of an interaction like this, I tend to break it down into 3 stages. The initial stage of mutual recognition, when the plant and bacteria identify each other, the establishment phase, when the structure is actually built, and then the functional stage, when the plant and bacteria are negotiating with each other and engaging in resource exchange. And although I'm not going to talk about the mechanisms underlying this very much, I just want to give you a sense of what's happening. So if I zoom in on a plant root, plant roots have layers of cells, and they have these little protrusions called root hairs that are a lot like the microvilli in our small intestines. Around those root hairs, you've got a bunch of soil bacteria, including rhizobia. And so what initially happens to kick off this interaction is that plants emit chemical signals into the soil called flavonoids. Those are recognized by bacteria in essentially what's a chemical conversation. And then those bacteria respond by producing something called a nod factor, which the plant can recognize. So this is essentially a chemical back and forth between the plants and bacteria that helps them identify that they've found the right partner, because this interaction is actually fairly specific. Then what happens is the plant root hair actually curls and traps the bacteria in a hollow tube that grows down into the center of the root. This is wild. So basically what the plant is doing is creating a mini culture tube in which it's going to propagate this bacterium that it's trapped. You see the nodule start to form, and this anatomically is what a mature nodule looks like, at least schematically. So inside the nodule, there are multiple zones where the bacteria and the plant are doing different things. The one that's most relevant for our purposes is the nitrogen fixation zone, which is when the bacteria are actively fixing nitrogen for the plant. So they actually become essentially intracellular plant organelles. They no longer can differentiate, they can't reproduce. They're just nitrogen fixation factories for their host. Someone else who works in the biology department at Penn, Errol Akçay, has done a ton of the foundational work on the economics of this particular interaction. So one reason I'm so lucky to be here at Penn is that one of my colleagues is a theoretician who works on exactly these questions. And so legumes and rhizobia, this mutualism in particular, has been used as a model, an economic model for species interactions. And when you read this literature, you see a lot of analogies and straight up stealing math from economics. And so in this paper, what Errol is doing is using a model that's analogous to collective bargaining between unions and firms to model the negotiation of the carbon for nitrogen exchange rate. You'll also see papers that use things like the Nash equilibrium, which is really commonly employed in economics as well. And so this is just a smattering of some of those examples. Generally speaking, what people in my field are interested in is how things like the context in which this mutualism is playing out influences the trade or the negotiated exchange rate. There's also some really beautiful recent work trying to dig into which partner exerts more control over the exchange rate and whether the negotiated exchange rate is fair or unfair. And there's even some work on sanctioning. So the idea that plants seem to be able to penalize or restrict carbon flow to uncooperative rhizobia. So when I said at the beginning that this is a marketplace, I meant it in a lot of ways, somewhat literally. A lot of the same things that we imagine happening in human marketplaces seem to be happening in these nodules. And one reason that this system is really easy to study is that the currencies are well defined: nitrogen from the bacteria and carbon from the plant. Okay, so what I said at the outset was that my lab is really interested in what happens when this process, when communication or negotiation between these organisms is intercepted or disrupted. And today I'm going to talk about 2 sources of disruption and interception. One is interception by other microorganisms like parasites or pathogens. The reason that I think this is a really interesting biological question is that, as you know, from work on, say, human microbiome studies, no organism is colonized by a single microbe. We are all ecosystems that host hundreds or thousands of microbes. And so what my lab is really excited to understand is how interactions with multiple different microbes at the same time might break some of these interactions. The other thing I'll talk about at the very end is how human environmental modifications like climate change or urbanization may also impact the outcomes of these mutualisms. Okay, any questions before I jump into our research system and some data?
You're gonna say a little more about how the bacteria can no longer reproduce. Or benefiting from this.
I'm actually not. That's a really great question. So if I go back to that diagram, there's a lot of work in the system on that question as well. So in this particular type of nodule, what happens is that these bacteria that are fixing nitrogen are what are called terminally differentiated. And so it's puzzling when I describe it like that, why these bacteria would fix nitrogen if this ends up being a dead end. It's thought that this is a really classic example of kin selection, essentially. And so what's not immediately obvious on this diagram is that there are also free living bacteria at the tip of this nodule that are close relatives of the ones that are domesticated inside of these organelles. And so we think that basically this operates by kin selection when this nodule senesces, the ones that have retained the ability to differentiate disperse back out into the soil. Okay, so the parasite that we work on is this thing called a root knot nematode that I had never heard of until I started reading some agricultural literature when I was a postdoc. It's in the genus Meloidogyne. A couple of things about it, and then I just want to show you some pictures of what it does. It's an obligate endoparasite, so its reproduction is tied to the plant. It can't reproduce without infecting a host. It's super common in agricultural systems, so we know a lot about it. It can infect thousands of host plants. My grad student, Matt Calvert, tells me that it has the broadest host range of any known parasite in the world. Is that right, Mac? Yeah, so incredibly generalist. It forms these structures called galls on plant roots. So like rhizobia, it's also forming these unique organs. And those organs are unidirectional flows of resources. So going to the parasite is obviously not giving the host anything. This is what the galls look like on a plant root. So I'm showing you tomato seedlings right here. You can see these little sort of like swellings or growths that look like beads on a string. This is why they're called root knot nematodes. It looks like the roots are tied in knots. If I zoom in on one of these galls, it looks like this. So you see the gall at the center of a bunch of roots. The nematode, you can't actually see. She's in the middle of the gall. What you see here is the eggs that she's laid outside the gall. And what you're seeing swarming it are all the little nematodes. So they're essentially little worms. I said she because only female nematodes form galls. There's a bunch of cool stuff about the sexual biology of these things that I'm not going to be able to talk about today. So this is the parasite that we're really interested in. And it's really amenable to some of the questions we want to ask because it forms a structure kind of like rhizobia does. And so it's easy for us to get some traction into some of these questions. And it's really easy to assay things like parasite load or mutualist load on a plant. Okay, so how might a parasite intercept or disrupt an interaction between a plant and a microbe? I'm not going to talk very much in detail today about the mechanisms, but I just want to give you a sense of at least sort of the three main ways I could imagine parasites, the mechanisms I think might underlie the patterns that I'm going to show you. So one is that these parasites could be intercepting signals or intercepting the conversation between the host and the mutualist. So this is a phenomenon you might be familiar with as eavesdropping if you've thought about signaling or communication literature, where a signaler, in this case the plant, is producing a signal that's destined for a particular receiver, in this case the rhizobia, and nematodes have the ability to intercept that signal. And so signaling for rhizobia could make it easier for parasites to find a plant. This is one way. The other possibility, or another possibility, and I'll show you data to support this in a second, is that nematodes could disrupt the establishment of nodules. So if you were to measure the number of nodules on plants with nematodes, you would find that they form fewer nodules than those without. And then the last possibility that my lab has gotten really interested in lately is that nematodes may actually be hijacking or modifying trade when they co-infect a plant, such that nematodes, so we know nematodes are stealing carbon, so sugars from the phloem. They're also likely stealing amino acids, which are nitrogen rich. So what's particularly interesting about this parasite is that it is probably drawing down both of the traded resources in this interaction. And you might imagine that that's going to influence things like the cost of engaging in this mutualism or potentially the exchange rate between hosts and bacteria as well.
So I have some quick questions.
Yeah.
But when you say intercept signal, I guess it could be two things. One is it detects the signal and comes and joins the fun. And the other is that it detects the signal and then blocks it so that the microbe doesn't actually come in and use things.
I'm imagining more the former, although I don't have direct data on either. It's possible, I'm trying to think what's consistent with the data that we have. It's possible that the latter could be operating, but the way that diffusible chemicals are working in the soil, it seems unlikely to me that nematode recognition of the flavonoids is going to decrease the rate that they get to the rhizobia. That's my guess. So I work on plants. I haven't actually showed you a picture of a whole plant yet. My lab works on this group of plants called burr clovers. Alfalfa is in this group. Alfalfa is Medicago sativa. So it's a legume. A lot of these are roadside weeds, essentially. One of them, the one in the center called Medicago truncatula, this one here, has been developed into a genetic model plant for studying some of these interactions. And so that's primarily the one that we work on, although we do some work in the field as well that I'll talk about if I have time. So all the data I'm going to show you today, it comes from Medicago truncatula, which is this plant in the center. Yeah.
Sorry, this is not your style, but
yeah.
Could you just stay a little bit closer?
Sorry. Yes. Sorry.
You're cutting out a little bit.
Yeah. Sorry, folks on Zoom.
There's 15 people there, so
okay. Thank you. Okay. So generally speaking, a lot of what my lab does is ask what happens when nematodes and rhizobia colonize the same plant. What happens when you put them together? And I'm just going to summarize my postdoc work on this topic in one slide before I jump into the stuff that my lab has done. So the first thing that you see, which is really exciting and puzzling, is that plants that form more nodules are more susceptible to this parasite. So we measured this by measuring the number of nodules on a plant and correlating it with the number of galls on a plant. What I'm showing you here are actually genotype means. So this is what's known as a genetic correlation. We know that this is not just driven by the environment. And we've also corrected these data for plant size. So this is not just purely a function of the fact that plants with rhizobia are bigger. So what this suggests to us is that there's something about forming more nodules that is associated with the formation of more galls. It suggests that engaging in this mutualism affects susceptibility to this parasite. The other thing that we found is that nematodes inhibit nodulation with rhizobia, and this is maybe our most repeatable result. So over and over again in different experiments, when we inoculate plants with nematodes, we find decreases in the total nodule biomass that these plants are making. And again, this effect is consistent even after you control for things like plant size. So essentially what we're seeing is a decrease in the relative investment in nodulation on plants that are infected by parasites. Okay, so I want to first start by focusing on this result, that nematodes seem to inhibit nodulation with rhizobia. One straightforward explanation or hypothesis for this pattern is that rhizobia nematodes are essentially resource competitors. So they're competing for access to, say, carbon on a host. And when you have competition, you expect to see mutual inhibition. So this one prediction you could make based on this hypothesis is that rhizobia should inhibit nematodes too, so that this inhibitory effect should be mutual. And so to test this idea, two people in my lab, Nuri Yi, who started out as a research technician and is now a PhD student, and Beth Vasquez-Smith, who is an undergraduate thesis student finishing up right now, set up to run an experiment where they measured the mutual effects of rhizobia on nematodes and nematodes on rhizobia when they colonize the same host. And I'm not going to go too much into methods in this talk, but I just want to give you a flavor for how we run experiments in my lab. Because for the most part, we're not doing genetic or genomic, we're certainly not doing genetic or genomic manipulations. A lot of what we do is inoculate plants with different things and see what happens. So in this experiment, what we did was inoculate plants with either just rhizobia or nematodes, or with both rhizobia and nematodes. This cohort that I'm showing you here were inoculated 4 weeks after they germinated. The reason I'm showing you this timeline on the x-axis is that we actually planted in cohorts to manipulate the age at which hosts first encountered these organisms. And the reason that we wanted to do this is that as hosts get older, we know that they change how they respond to microbes. One really great example of this that you're probably familiar with in humans is that kids tend to be more susceptible to most infections than adults. It turns out that that pattern is true in plants as well. Susceptibility to disease of juveniles is a super common and repeatable pattern even outside of humans. So that's why we wanted to be able to capture an effective host age if there was one here. So this experiment was really large because what we wanted to do was get snapshots over time of how these processes were playing out. And so we harvested these plants in cohorts that are indicated by these white circles. Today, I'm just going to show you data from this last cohort towards the end. So what you're going to be looking at are data on plants that were harvested 7 weeks after inoculation. And those plants are going to vary in the age at which they first encountered these parasites or mutualists. And so I'm going to show you these data like this. I have my proxy for parasite infection, number of galls on the left, my proxy for mutualism, for symbiosis, number of nodules on the right, and then I have host age, the age that this host encountered this critter on the X. And I want to start just by showing you the data that I showed you before, evidence that nematodes inhibit rhizobia. And so if we compare plants that were only inoculated with rhizobia to plants that were also inoculated with nematodes, we see a decrease in nodulation. What was really interesting is that when we look at the effect on the parasite, we see the opposite result. So rather than seeing mutual inhibition or competition between these two critters, we actually found that rhizobia seemed to be facilitating parasite infection. So when you only inoculate plants with nematodes, they form fewer galls than when you also give them rhizobia. So I just want to take a second here to say that this is arguably the worst case scenario if you're the host, because the parasite is inhibiting the mutualist and the mutualist is facilitating the parasite. So it's a really intriguing pattern to me as an evolutionary ecologist because it suggests that there are pretty massive fitness consequences of this conflict for the host. What was even more interesting to us was what happened when we looked across different host ages. So how old the plant was when it first encountered rhizobia and nematodes. And what we see is that these facilitative and inhibitory effects only occurred in hosts that first encountered both of these critters at older ages. So when hosts were inoculated at only 2 or 3 weeks old, we don't see any competition, so inhibition or facilitative effects. Mechanistically, I don't know exactly what's going on here, but it suggests to me that whatever mechanisms underlie these interactions between rhizobia and nematodes must be changing as the host ages, because what plays out on a 4-week-old host is different from what plays out on a host that encounters these critters when it's much, much younger. So this is all I'm able to tell you about this result right now. I'm really interested in following it up, for example, by tracking host gene expression over time to ask what's changing about either the distribution of resources or immune functions as hosts age that might be influencing sort of the crosstalk or conflict between these two organisms. What I want to focus on next is this facilitative effect of rhizobia on nematodes. And the reason that I want to focus on this next is that symbiotic microbes often, but not always, protect against parasites and pathogens. And so if you've encountered any microbiome literature, either in the primary literature or in the popular literature, what we often hear about is the potential for microbes to protect us from infection. So protect us from pathogens, protect us from parasites. Rhizobia are doing exactly the opposite in this system. So there's something about this particular mutualism that's making the host more vulnerable to infection, not less vulnerable to infection. And so we wanted to chase this down a little bit and ask why symbiotic microbes might increase susceptibility to parasites. And so here I'm going to talk at least a little bit more mechanistically about how sort of crosstalk or conflict between these two interactions might be occurring in the plant. This project was also led by Nuri when she was a research technician in my lab. She's currently working on writing this up as a first authored manuscript now that she's a PhD student here. So Nuri and I, when we were sitting down to tackle this question, formulated two potential hypotheses that could explain this pattern. One is that forming a symbiosis might increase parasite infection in and of itself. And this is related to what I told you about sort of the role of signaling and establishment in symbioses. So establishing a symbiosis, for example, signaling to or attracting your symbiont might attract parasites. We also know that when hosts are establishing symbioses with microbes, they have to shut down local immune responses. So it's also possible that a cost of that is infection by a parasite or a pathogen. But the other hypothesis that we had focused more on the economic features of this mutualism. We thought it was possible that symbiont provided resources, or in this case, nitrogen, might increase parasite infection. And the reason for this is that we know in, for example, the plant herbivore literature, that herbivores preferentially attack plants that are nitrogen rich because it's a more nutritious meal. And so these two things are confounded in most of the experiments that we've done because rhizobia form nodules and provide nitrogen at the same time. So what Nuri did was set up an experimental test that at least partially decoupled the establishment of a symbiosis from the benefits that it provides. And this is easy to do in this particular mutualism because it's easy to manipulate nitrogen outside of the rhizobia mutualism by giving plants fertilizer. So if you buy over-the-counter fertilizer like Miracle-Gro, nitrogen is one of the three main components of it. You'll see it described as NPK ratios, and the N is nitrogen. So what we could do in our lab is mix up our own batches of fertilizer where we control the amount of nitrogen and then ask whether increases in nitrogen translate to increases in parasite infection. So I just want to quickly show you the predictions that we would make under these two hypotheses. So if forming a symbiosis independent of nitrogen is what drives susceptibility to parasites, then we would expect to see a difference in parasite load between plants inoculated with rhizobia and those that are not inoculated with rhizobia. So those that have symbionts and those that don't. If what we're seeing is an effect of nitrogen, we would expect to see no difference between plants with and without rhizobia, but a strong effect of how much nitrogen we give the plants. Okay. So I'm going to show you the data in a similar format to the toy graphs that I just showed you. The one thing that I want to mention before I show you these data is that we actually have 4 different symbiosis treatments, not just one. We have one group of plants that were uninoculated, and then we performed this experiment with 4 different rhizobia strains. And the reason that we did this is that different rhizobia strains differ in their effects on the plant. So we wanted to make sure that we were capturing relevant variation in what the symbiont was actually doing. Okay. So what we see is that nitrogen seems to increase susceptibility to the parasite, and symbiosis alone, having rhizobia, had no effect or a very weak one. So what you see is increases in parasite load in these 2 treatments that had more nitrogen. And the difference between these gray points, which are the uninoculated plants, and those that are inoculated, are approaching statistical significance, but are not significant. So my interpretation here is that the effect of symbiosis seems to be much weaker than the effect of resource provisioning. Or in other words, that the connection between this symbiosis and parasitism might be via the traded resources rather than via all of the corresponding physiological changes that happen in a plant.
The parasites, are they able to get nitrogen out of the soil alone, or they can only do it when they're in a gall?
Great question. As far as I know, they can only do it in a gall. They're the juvenile forms, when they hatch out of the eggs, I'm pretty sure can't feed on anything in the soil. So they just crawl towards a plant and then feed once they establish a gall. Okay. So one problem when you're talking, or one complication, I should say, not a problem, when you're talking about resource manipulations, is that resources are affecting a whole bunch of things about these plants that have nothing to do with parasites. And the major one that you might imagine is that plants grow much larger when nitrogen is abundant. So any of you who are gardeners would recognize that this is what happens when you put fertilizer on a plant. And so what we wanted to do to account for this was to size correct our parasite loads. So we have these major differences in overall plant size across these treatments. What we want to know is what happens to parasite load relative to overall size across these different treatments, because having, say, 5 parasites on a tiny plant is very different from having 5 parasites on a large plant. And so when we do this, we see a really different pattern. Now we still see an effect of nitrogen, but not symbiosis. So we're still seeing that resources seem to be the driver of susceptibility to infection. But now we see that parasite load is peaking at intermediate nitrogen levels. And so we see very low parasite loads at low and high resource levels, but super high, relatively high susceptibility at intermediate levels. Okay, so overall, at least in the context of this symbiosis, it appears that the effect of forming a symbiosis on parasite infection is much smaller than the effect of resources. So this suggested to us that the role of trade and the role of resource exchange might be a really important factor linking these two interactions. It implies that engaging in resource exchange might increase vulnerability to parasites more broadly. In other words, that this is something that we should think about in other resource exchange mutualisms that, say, humans or other animals or plants engage in. But it also told us that this effect is complex, that the protective versus vulnerable effect of resources can vary depending on the absolute level of resource abundance in the environment. The other thing that was really surprising to me about these data is that there wasn't a strong effect of symbiosis formation in and of itself on this interaction. And to be honest, this is exactly what I thought we would find when we started this experiment. I thought we were going to see that inoculating plants with rhizobia was the major thing that drove susceptibility to infection because of all of the work on immune crosstalk between mutualists and parasites and pathogens. But at least in this particular system, that doesn't seem to be the major effect that we're detecting. And so I think an interesting question is why? How are hosts mitigating the potential pleiotropic effects of these two symbioses on each other? Okay, so to summarize sort of the highlights of what I've told you so far, rhizobia seem to increase susceptibility to nematodes, and our data suggests that it's partially driven by resources. I've also shown you that nematodes disrupt nodulation. But all of the data, or most of the data that I've showed you so far, has just manipulated rhizobia presence or absence. Ecologically, that's not a realistic thing to do at all, because if you dig up any legume, they all have rhizobia. You're never going to find a plant in the field that doesn't have a nodule on it. What is relevant is that we know that these legumes can partner with many different strains. And the experimental manipulation I showed you a couple of slides ago sort of implies this, that there are a bunch of different strains you can inoculate onto a plant. They can all form nodules, but sometimes the outcomes are different. And in fact, from other work, including work by my grad student at Mac, we know that strains differ a ton in the amount of benefits that they provide. So it suggests that strains may actually be strain variation may be a really important factor to consider when we think about crosstalk between parasites and mutualists. So I just want to show you a slide of Mac's really interesting recent data to bolster this point. So Mac was really interested in the sort of variation among rhizobia strains in the benefits that they have for plants and in the resource exchange economics that emerge from that interaction. He has a ton of really cool data I don't have time to show you, so I'm going to summarize his results in 2 graphs. The take home from both of these graphs is that rhizobia strains differ in the benefit they provide to their host and in their broader effect on plant resource traits. So I'm showing you 2 of the traits that he measured to capture this. One is the total amount of nitrogen that plants accumulate in their above ground tissue. He did this experiment with 2 different plant genotypes, but what I want you to focus on are the different rhizobia strains, which are the different colors in this graph. And what you see is that plants accumulated more nitrogen in above ground tissue with one strain than they did with the other, suggesting that they're reaping a larger benefit from one. The other thing that he found that I think is actually even more interesting is that rhizobia strains seem to be affecting plant photosynthetic rates. And so for those of you who don't think about plants very much, the reason photosynthesis is relevant is that photosynthesis is how plants make carbon. And so what this is telling us is that these strains are also potentially impacting the rate of carbon fixation in these plants. So they're impacting the influx of carbon into the system as well. And so this told us that considering strain differences or variation among different potential partners might be really informative to understand what's going on in these interactions. And so the last set of projects I want to talk to you about on this theme tackles this question, asking whether plants with different rhizobia strains respond differently when they encounter a parasite. And so what I'm essentially asking here is whether partnering with a different rhizobia has cascading effects on the other interactions that this particular plant is engaging in. And again, this is motivated by the fact that we know in other contexts, rhizobia have hugely different effects on plants. What we haven't thought about as a field is how those effects translate to interactions with parasites and pathogens. And there are a couple different ways you could imagine asking this question. The way that we decided to do it in this case is to measure plant gene expression in nodules and galls. And so what we're doing here is zooming in to the interface of the interaction between the host and the mutualist and the host and the parasite, and asking what differences we see in those structures when plants are partnered with different rhizobia. This is one incredibly powerful thing about this particular experimental system, is that these interactions are conveniently packaged into two different structures. And so we can grab a nodule and get a proxy for what's going on with the mutualism, and grab a gall and get a proxy for what's going on with the parasitism. You couldn't do this with something like a systemically infecting pathogen because you wouldn't be able to isolate that interaction. So this is an experiment that is led by Addison Buxton Martin, who's a PhD student in my lab. He's collaborating with Emil Glucktaler, who was an old postdoc in my lab, and Nuri, who was the research technician when this experiment was run. And so I'm going to show you two sets of data to address this question. The first thing that we wanted to ask was whether rhizobia strains change plant gene expression in galls. So if you look at a gall, if you look at the genes that are turned on in a gall, do you see different genes turned on and off when plants have rhizobia A versus rhizobia B? For this experiment, the two rhizobia strains we used were both strains of a species called Ensifermella lodi. We used a strain called 1021, which is notoriously a poor nitrogen fixer, and 1022, which is a good nitrogen fixer. So I'm going to show you the data in a graph that looks like this. So on the x-axis, I'm going to have the effect of rhizobia on gene expression in nodules, as measured as the log-fold change between the two different rhizobia treatments. On the y-axis, I'm going to show you the effect of rhizobia on gene expression in galls. And so just to help you keep track of that, I'm going to put these words on here. Before I show you the data, I want to show you what we would expect under a couple of different sort of crosstalk scenarios. So one possibility is that the effects of rhizobia on plants are confined to nodules. So these effects are really strong in the structure that rhizobia form, but they don't really propagate out to the rest of the plant. And if that's the case, you would expect to see the data in this figure cluster here, where we see large strain effects in nodules, large log-fold changes when we look at genes expressed in nodules, but no effects of rhizobia when we look in galls. What I should have said is that each point on this graph is going to be a gene. So what we're going to look at is where different genes fall out in this space. We could see that strains have similar effects on gene expression in nodules and galls, so that whatever changes rhizobia strains cause in nodules, you also see propagated into this parasitic structure. And then the third possibility is that strains could have opposite effects in these two structures. So what a strain does in a nodule is totally different from the cascading effect it has on the parasite. So when I show you Addison's data, what he found is that, first of all, rhizobia are having really strong effects on plant gene expression in the parasitic structure. So this is sort of, yeah, if I think about how to frame this, we've been talking about this as a cascading effect, because what's happening is the rhizobia are affecting the plant, which are then affecting the parasite. Something that you may not appreciate, having not looked at these structures on plant roots, is that they're physically separated in space. So this is also an effect that we think is cascading through the host. So what we see is that most of the genes that are responding to rhizobia are responding similarly in nodules and galls. We do, however, see a couple of interesting genes that are shown in green here that seem to have opposite effects in nodules and galls. So if they're upregulated in the context of the good nitrogen fixer in a nodule, they're downregulated in a gall, or vice versa. So in other words, they're responding oppositely. The reason that this effect we think is really interesting is that it can't really be predicted or explained by just a straight cascade through the entire plant. There has to be something specific going on in the gall that would be explaining this particular outcome. So the second thing Addison asked was whether nodules with different resident rhizobia strains respond differently to parasite infection. And so to do this, what we did was compare gene expression in infected and uninfected plants, in nodules that had one strain or in nodules that had another strain. And so what I'm going to show you here on the x-axis, this is a measure of the effect of parasite infection in nodules with 1022. The y-axis is a measure of the effect of parasite infection in nodules with 1021. And we can sort of similarly, just to orient you in space, think about what it would mean if genes fall out in different regions of this space. So if we see a pattern that looks like this tells us that nodules with different rhizobia strains respond similarly to parasite infection. Or in other words, that rhizobia aren't really impacting how a nodule responds to infection. If we see something like this would tell us that nodules with different strains are responding really differently to parasite infection. And to our total surprise, we see the latter pattern. So we see that nodules with different resident rhizobia respond really, really differently to parasite infection. And so what Addison is showing here are genes that have a significant strain by infection interaction, which means that they statistically responded differently in nodules where 1022 was resident and nodules where 1021 was resident. So to me, this is kind of wild because it suggests that the partner that is resident in a nodule is mediating the effect of a parasite on whatever is happening in that nodule. It also suggests that parasites are impacting gene expression in nodules a lot, which again is consistent with the idea that they might be impacting trade or other outcomes of the mutualism. So Addison has done what are called gene ontology analyses on these genes to try to identify their functions. I don't have time to talk about them today. What I'll say is that we do see some suggestion that functions related to trade are involved, but that's all I have time to say now. Okay, so overall, what these data suggest is that plants with different rhizobia strains respond really differently to parasite infection, and that the partner that you have seems to actually be a surprisingly important factor in mediating your interaction with this antagonist. This is because these plants are expressing different genes at the site of parasite infection itself, so in the gall, but also that the effect of parasites on nodule gene expression seems to be dependent on strain. So what I want to do now is just wrap up what I've told you and give you a quick glimpse into the two new projects that my lab is starting. So overall, I think our data suggests that parasites are disrupting both the formation and function of this major host microbe symbiosis. By formation, I mean formation of nodules, so the number of nodules a plant has. By function, I mean gene expression profiles of those nodules. Our data also suggests that symbiotic microbes seem to be making their hosts more vulnerable to infection, in part because of the resources that they provide. So that resource economics might be linking these two things together. Okay, so there are two new threads of research in my lab that I'm really excited about that also tackle this umbrella question from a really different perspective. So in these two new projects, we're really interested in what happens when cooperation or negotiation are disrupted, not by another microorganism, but by some feature of the environment that's changing. And the two environmental gradients that we're working on are climate change and urbanization. So I'm just going to show you one slide on each of those to justify the connections and why I think they're so cool. So as all of you know, temperatures are rising rapidly around the world. So we expect temperature changes to be a major environmental factor that these mutualisms are going to encounter in the future. What is really cool about this particular interaction is that we know that both the production of nitrogen and the production of carbon are sensitive to temperature. So the rate at which those resources are produced is a function of temperature. And so what that suggested to us is that changes in temperature could affect the outcome of trade in this particular interaction by modifying either the rate at which these resources are produced or the negotiated exchange rate. And so what we're doing is using elevational gradients in the Southern Appalachian Mountains to start to test some of these ideas by sampling plants from valley populations, which are warm adapted, and mountaintop populations, which are cold adapted. The second environmental feature that we're thinking about is urbanization. So I have the Philadelphia skyline here because this work takes place in Philadelphia. And the reason that we're interested in this is that this recent paper captures it best. They describe the phenomenon of global urbanization as an unintended but highly replicated study of global experimental evolution. And what they mean by that is that if you go to any city around the world, the environment changes in basically the same way as you move from the city center to rural outlying areas. And so you have this beautiful replicated environmental change that you can use to ask questions about how those gradients impact the function of the organisms that live along those gradients. And so for this project, what we've started to do is sample plants and microbes from along an urban to rural gradient in the Philadelphia area. This is actually something that we partnered with Linda Robinson, who's an instructional lab coordinator in the Department of Biology. And this is the basis of a course-based undergraduate research experience for intro bio. So these students are working with the rhizobia strains that we've collected in urban and rural Philadelphia, inoculating them onto plants and then measuring differences in their effects. And this project is being led by Terrence Topping Brown, who's an incoming PhD student in my lab, and Chang-Yu Chang, who's a postdoc. And so with that, I want to thank everybody, all of my lab collaborators who did and led all of the work, everybody who supported it, and then you guys for coming to hear about plants and microbes in a MindCORE seminar. Thank you so much.
So right at the end, I'm not going to get very surprised by it, but you suggested that the rate of carbon-nitrogen exchange gets modulated. Is that something that the differences in gene expression you see in the nodule produced by the nematode might be driving? I have kind of a simple-minded idea that more nematodes that are coming, less nitrogen you want to be paying for, or something like that.
Yeah. So I think that's possible. We don't have data that can address that directly right now. There are a couple ways I could imagine it playing out, actually. One is the way that you described. The other is that depending on exactly how nematodes are impacting like the influx of carbon and the outflow of nitrogen, I could imagine increasing nitrogen fixation rates as well. So that project is something, an experiment I didn't get to talk about today because we haven't analyzed the data yet, was to measure resource exchange rates in uninfected and infected plants to compare them and try to get at exactly what you're asking. So do things like compare like the total amount of nitrogen that ends up in above ground tissues in both of those contexts. The other thing that I would really love to do is work with Errol, who's the modeler of mutualisms, to modify some of the models he's made of this interaction to incorporate a parasite and ask that question theoretically. So that's something he and I are talking about as a component of the climate change project as well. Yeah. Yeah.
With experience of like counting a number of normalizing by the
Yeah.
how do you start to think about what is the right normalization?
Oh man. Yeah, so it's such a good question. So and it's a huge question in the biology of parasite infection in particular, because size correction is such a big, it impacts your results a lot. What we've done historically is just correcting for mass, because that's what's easiest to measure, because you can weigh the root system. A project that Chang-Yu, the postdoc in my lab, is working on doing is training machine learning algorithms to measure root system size and count nodules and galls automatically. And that I think would be a better proxy, because I actually think surface area would be a better measurement of size. The other thing that we know is that nematodes infect plants through the root tips. So those algorithms could actually count root tips potentially, which might be an even better metric. And the last thing that is really important about your question is that nodule size varies a lot. So a lot of what we do is just count nodules because it's easiest. because plucking nodules off roots and weighing them is impossible for most experiments. That's another reason that this machine learning approach would be really great to get up and running, because we could measure nodule area, correct it for plant surface area, and I think get a much better measurement of like relative loads. So that's a very long-winded answer, but it's a question we're thinking a lot about right now. Yeah.
Thanks for an interesting talk. You may have sort of talked about this a little bit, I just missed it, but can you say a bit more about the sort of urbanization study that you have planned and
Yeah.
sort of what your predictions are there?
Yeah, absolutely. So I didn't say it. So there's been some work out of a lab at the University of Toronto on how urbanization affects this particular mutualism. What they've done is just go out into the field and count nodules on plants along urban-rural gradients. And what they see is let me see if I can get this right, a decline in nodulation towards city centers. And their hypothesis is that in city centers, there's a ton of nitrogen-rich runoff from roads. Something I didn't go into, but is a whole other major avenue of work in this field, is the impact of external nitrogen inputs on this mutualism. And when it's abundant, plants typically don't nodulate rhizobia because they can just get nitrogen directly out of the soil. So what we would expect to see, my prediction based on their work, is that we would expect to find less cooperative rhizobia in city centers, rhizobia that provide fewer benefits, because that relaxes selection on cooperation exerted by the plants. So that's what we're expecting. What I think is shaking out, so this was run, the first round of students ran a pilot version of this last fall, and it just went through the full intro bio class this spring. So I haven't looked at those data yet. What it looks like is that there's also a lot of variation among urban sites and among rural sites. And so it looks like we might need to think a lot more about like micro environmental variation within each of these regions too.
Is the runoff from like vehicle traffic or what?
Yeah, so either that or lawn treatments. So the way that we collected these things, they're sidewalk weeds, basically. So one of our collection sites was along the fence at the Penn Medicine Septic Station. We collected from the trolley portal. Out in the suburbs, we collected from along sidewalk city park boundaries as well. So what we were trying to do is collect from similar physical environments in those two different spaces that would be experiencing a whole bunch of different inputs from whatever else. Yeah. Yeah.
You have a plot showing something about temperature and it looked like the, right, okay.
Yeah.
So it looks like the effects are similar, right? So the nitrogen effect from temperature and the photosynthetic effect from temperature are following a similar course.
Mm-hmm.
So that seems like it's not what you want for evaluating the market effects of these.
So what's hard to see on this particular graph is that they're measuring these effects in a bunch of different plant species and across a bunch of different growth temperatures. So what's hard to do on this particular figure is compare the same species in the same conditions. And I glossed over this, but it's a really important point. When you do that, you find that these curves are offset. And that's the piece that got me really excited about this, because if they are offset, then you would expect temperature changes to change the relative rates of production of these two things.
Yeah.
So it's not at all obvious from this. What we're doing this summer, so I'm going to be in the field for 2 months this summer. One thing I'm going to be doing is collecting preliminary data for this proposal across an elevational, like a 2,000-foot elevational gradient, to confirm that we're seeing what we expect. Yeah.
So,
Mhm.
so you want to observe and you not believe that the plants at different age, right? You see that the infections are dependent on the age of the plant. So I was wondering if those plants at different age also have a different size, and size plays a role in that as well.
Yeah, great question. So those data are among the few that I did not size correct. And the reason for that is that those plants have encountered they were different ages when they first encountered their symbionts, but they've had them for the same length of time. And so we don't a priori expect differences in number of nodules as a result of age because they've had them for the same length of time. That being said, the next thing on my list to do is size correct those data once we get the roots weighed, just to make sure that we don't see qualitatively different patterns once we do size correct them. Because the plants that were older when they encountered rhizobia nematodes were also larger. So we would want to eliminate the possibility that there's some sort of dynamic interaction between those two processes. It's a really good question. Okay, thank you guys so much.